Okay, ladies and gentlemen, good afternoon. As you can see from the slide, my name is Mukhtar Ali. So today we are going to look at the area of immobilized enzymes and their applications. So class, the essence of this class or the essence of this tutorial is very important. Why? Because you know that when you are trying to isolate an enzymes, there is a lot of hurdles, pros and cons, and there is a lot of economics burden. So you see, it means that after you purify your enzymes, when you want to use or when you want to get that the same enzymes, you need to follow the same isolation techniques. So that is why now the scientists think of coming up with a new way of, after you isolate the enzymes, you can immobilize it in a system that you can use it and reuse it again. So that is the essence of immobilized enzymes or immobilizing the enzymes. When you immobilize the enzymes, you can use them over and over again. So the essence of immobilizing the enzymes is to use and to reuse of your enzymes after you isolate it. So that is why, because of the economics burden, time consumption, and a lot of issues, that is why scientists now think of developing a system that will allow the researcher to immobilize the enzymes. So when you said immobilizing the enzymes, it means that you want to confine, you want to restrict the movement of the enzyme so that you can use it and reuse it again. So that is what we are going to look for the today's lecture. So now, without wasting our time, we are now going to go deep into the slides. So you see, these are the contents, which includes the method for immobilizing the enzymes, the applications, and the applications of the immobilized enzymes include analytical techniques, therapeutics, biosensor, and industry. Then the last we have reference, conclusion, and then references. So what is immobilized enzymes? So enzymes immobilization may be defined as a process of confining the enzyme's molecule to a solid support over which a substrate is passed and converted to products. So you see, as I mentioned, the enzymes, the immobilized enzymes or the immobilization is, you will try to get a solid support or a matrix or a system that will allow you to confine the enzymes on the support. So that is just it, because if you immobilize the enzymes, what you need to do is, when you are trying to produce your product, you just pass the substrate toward the immobilized system, then it will just be converted into the product of interest. Then you recover your immobilized enzymes and you can use it over and over again. So that is the meaning of immobilized enzymes. So you confined or you restrict the movement of the enzymes on a support so that you can just pass your substrate on the immobilized system, then it will be just converted into products. So that is what we call immobilized enzymes. Then the next thing is, why immobilize enzyme? Why do we need to immobilize the enzyme? So number one is protection from degradation and deactivation. If maybe the enzymes is allowed, or maybe it has just the potential to move from any direction, so the enzymes can be degraded and be denatured at the same time. So that is why it's very important to confine it, so that to protect the degradation and the activation of the enzymes. You know that if the enzymes are degraded or deactivated, it means that they are not going to carry out any of their enzymatic activity. It means that they are now waste. So that is why we need to immobilize the enzymes to prevent the degradation and the deactivation of the enzymes. And number two, the reuse of enzymes for many reaction cycles. So you see, you can, as I said, you can use the enzymes and reuse it over and over again. So you can use it for as much as many reaction cycles. and as a result of that, it of course leads to the lowering of the total production cost of the enzyme associated reactions. You see that maybe if you use your enzymes once, because of the hurdles and the pros and cons of isolating the enzymes, you have to follow the same procedure before you get that the same enzyme. So that is why if you are not actually immobilizing enzymes, even in the industry and in pharmaceutical industry and many areas in life, you can actually find the enzymes or the products of that particular enzymes to be highly positive. So that is why we immobilize it, so that we can use it and use it again, so that we can actually lower the production cost of our products. That is number two. Then number two, ability to stop the reaction rapidly by removing the enzymes from the reaction solution. You see, actually when you have your enzymes, immobilized enzymes, then you have your substrate. Once you realize that your enzymes is or was able to produce your product of interest, then you just remove it directly. You just remove the immobilized enzyme. Then from there, then you actually save the efficacy and the efficiency of the enzyme. Because truly speaking, one important thing that I want you to understand is that when you are using immobilized enzyme, the more you use it, then you are actually reducing the efficacy of the enzyme. So that is why it's very important. Once the enzymes catalyze your products or completed the reactions, then you need to actually remove it, so that to try as much as possible to maintain the efficacy of the enzymes. Then the next thing is also have in enhancing the stability of the enzymes. You know that enzymes are very heat labile, temperature labile, and cooled labile, and other actually conditions like concentration, pH. So that is why also immobilizing the enzyme is very important, because it's trying to enhance the stability of the enzyme. So it also have in easy separation of the enzymes from the products. So you see, once the product is formed, then you can just simply separate your enzymes from the product, and that is why. So as a result of that, you can actually prevent the product contamination or product inhibition from normal metabolic pathways. Okay, so that is the reason why we need to actually immobilize the enzymes. Then these are actually this thing is not actually supposed to be here, but uh, this slide is just trying to show like what will give you the choice of selecting a matrix, because the matrix actually have different property and have different cost effectivity. So that is why there are some important parameters that you need to check, that you need to confirm before selecting a carrier matrix to use for immobilizing the enzymes. Number one is inert. When you said inert, it means that you have to make sure that your carrier is not reacting with your enzymes. And whenever you said inert, it means that something that does not undergo any reaction, it does not participate in the reaction. So you have to make sure that your carrier matrix is inert. That it means that it will not be reacting with your enzymes. Then second, physically strong and stable. You have to make sure that then cost effective, then regenerable in reduction in product inhibition. So also this one are very important in trying to choose the matrix. Then classification of carriers. We have the classification of carriers. We have three of them. We have inorganic carriers. So some of these carriers are inorganic. When you say inorganic, it means that they are not from living source. So you see examples of them. We have silicon oxide, porous glass, silica. We also have minerals, materials. Then we also have uh, lights. So you have, these are the examples of the inorganic carriers. Then we have organic carriers. We have like cellulose derivatives. We have like diethyl amine ethylene cellulose. We have uh, CM cellulose. We have dextran. We have polysaccharide, which comprise like agarose, starch, pectin, and chitosan. So these are the examples of organic carriers. So we have organic synthetic carriers. That is the carriers that you can just go to the lab, you synthesize them. Why do you do that? Because there are some enzymes that inorganic carriers and the organic natural carriers cannot actually be immobilized with them. So that is why the scientists now think of maybe trying to model or trying to design a synthetic one that can actually perfectly fit with your enzymes of interest. And then so these are actually like a processes or method of enzymes immobilization. Even though they are classified into two, we have physical method and we have chemical method. Like for example, examples of the chemical method, we have like covalent bonding. So then for the physical method, we have like adsorption, we have encapsulation, and we have entrapment. So we are now going to look at them one by one after the other. So these are the physical methods. We are starting with the physical method. The number one, we have adsorption. Adsorptions, it involves the physical binding of the enzymes on the surface of the matrix. So you see, this is your uh, carrier. Examples we have like silica, bentonite, and cellulose are the examples of the carriers that we use in the adsorption methods. So this here represent the enzymes. So you see, this is the enzymes and this is your matrix. So the process of adsorption involves the weak interactions like van der Waals attraction or hydrogen bonding. So you see, this it means that this is your enzymes and this is your matrix. So the interaction between the two is by weak interactions, which may be van der Waals force of attractions or hydrogen bonding. So that is this one. So examples of the enzymes that can be immobilized using adsorption method, we have catalase and invertase. Okay, then this is the advantage of adsorption method and disadvantage. Number one is simple and economic. Limited loss of activity, can be recycled, regenerated, and reused. So these are actually disadvantage. Relatively low surface area for binding. So you see that sometimes it means that their enzymes may not actually find the perfect area to bind on the surface. Then we also have exposure of enzymes to microbial attack. Yes, of course, because it's of they are all of organic molecules, so the enzymes can decide to be because of some microorganisms can excrete some proteases, so it can easily digest both of the enzymes and the matrix, so they are easy to expose to the microbial attacks. So that is the one of disadvantage. Yield are often low due to inactivation, and so these are just disadvantage and the advantage. Okay, then the next thing is entrapment. It's also a physical method. So for the entrapment, the enzymes or cells are not directly attached to the support surface, but simply trapped inside the polymer matrix. You see, so these are this polymer matrix. This one, are you seeing it? So this is the cellular matrix. So you see, this is your enzymes, the yellow color. This one, but this is your enzymes trapped in the matrix. So you see, they are all trapped. So this is so actually for the entrapment, it's not that the enzymes is binding on the surface, rather is entrapped inside the matrix. So the enzymes are held or entrapped within a suitable gels or fibers. So you see, this one is also enzymes entrapped. You see, this is your enzymes. Okay, then it is done in which a way as to retain protein while allowing penetration of substrate. It can be classified into lattice and microcapsule types. Okay, so you see, this one is trying to tell you that uh, it allow the substrate to pass here so that it can just easily convert it into your product of interest, and it's divided into two. So inclusion in gels, we have like these are the examples. We have polyacrylamide gel, polyvinyl alcohol gels. So we have cellulose and so inclusion fibers. So these are actually the matrices that are used in enzymes entrapment. So you see, as I said, it's divided into lattice type entrapment. So lattice entrapment involves the enzymes within interstitial space of a cross-linked water-soluble polymer. So you see, these are the cross-linked water-soluble polymers. So some synthetic polymers such as amide, we have polyvinyl alcohol, and natural polymer such as starch. So they are used in lattice-type entrapment. So you see the enzymes is entrapped within the interstitial spaces. You see, this is the interstitial spaces. So you see interstitial. So lattice. Then we have micro encapsulation. That is encapsulation. It involves enclosing the enzymes within semi-permeable polymer. You see, the other one is interstitial. So, but this one is permeable polymer membranes. So you see, this is how they are. So they are not interstitially. You see, all the enzymes are just encapsulated and just a single polymer. So examples of the semi-permeable of them, we have like nylon membranes, we have like colloidal. Okay, then these are the advantage: no chemical modification, relatively stable forms, easily handled, and reusage. The enzymes might leak from the force. Yes, that is the problem a bit. You see, the enzymes can leak from it. So that is for this one. Then we have covalent. So covalent is a chemical method. It's a chemical method. So based on the binding of the enzymes and water soluble carriers by covalent bond, the functional groups that might take part in this binding are, so you see, there are actually some functional groups on the matrix that react with some other functional group from the enzyme. So it's a reaction. So that is why we call it a chemical method. So some of the chemical bonding from both the enzymes and the matrix, we have like carboxyl group, sulfurhydryl group, hydroxyl group, imidazole group, phenolic group, diol group. So this is it. So, and the disadvantage: covalent binding might alter the conformational structure and the active center of the enzymes. Yes, seriously speaking, because sometimes at the active center of the enzymes we have amino acid residues. So if the amino acid residues react with the functional groups from the substrate, it's, of course, it's it will just change the conformation of the enzymes, meaning that the substrate cannot even find a position to bind to, so therefore no any catalysis will take place. So that is one of the disadvantage. Then advantage: the binding force between the enzymes and the carrier is strong. Yes, because it is a chemical reaction, so therefore you should not expect the leakage of the enzymes. So even in the presence of substrate or solution of high ionic strength, so it's very strong. Then we also have cross-linking. So in the cross-linking, look at it. Cross-linking involves intermolecular cross-linking of enzymes molecules in the presence or absence of a solid support. So it's just the enzymes. Is in this case, the enzymes can also rearrange themselves to cross-link with one another. But so also at the same time, the presence of the solid supports, it may also cross-link themselves together. If you look at them, look at it. This one bind with this, so they cross-link. So the method produces a three-dimensional cross-link enzymes aggregated, so by means of a multifunctional reagent that links covalently to the enzyme's molecules. So this is the cross-linking. So these are the advantage of cross-linking: very little disruption, that is, enzyme strongly bind, so it means that it's strongly bind, so there is very little disruption. Then it is high stable. Then disadvantage of cross-linking: you see, cross-linking might cause significant changes in the active site, so not cost effective. So you see, this is the disadvantage. See, this is just briefly on how, you see, pictorial presentation of different immobilization methods. You see, this is encapsulation. That is in the situation where enzymes are entrapped either interstitially or on a membrane polymer. Then we have covalent. You see, this is the covalent. So we have the enzymes, look at it, and this is the carrier. So they bind covalently through the functional group bond. Remember covalent catalysis, okay? Then this is adsorption. So this is not a reaction, it's just through a weak intermolecular forces of attraction, like Van der Waals and hydrogen bonding, and this is the cross-linking. So this is you see this is the interstitial, you can clearly see it. You see this is encapsulation. So limitation of enzymes immobilization. So generally, cost of carriers and immobilization, the costs, it actually very high cost effective. So changes in properties, yes, as a result of some binding and reactions, it may actually lead to changes in the selectivity or the properties of the enzyme, because it may be denatured, it may actually change its conformation and other things. Then we have mass transfer limitations. When we said mass transfer limitation, it means it's a condition where your substrate needs to have a direct contact with your immobilized enzymes. So it means that when trying to carry out the reactions, it means that the substrate must move to a location where your immobilized enzymes is localized. So therefore there is this problems of mass transfer limitation. It's not necessarily in all cases. Your substrate may not always move to the locations where you have your immobilized enzymes. So as a result of that, the reaction may not actually take place. I hope you understand. So that is why sometimes we need to actually develop what they call a bioreactor. A bioreactor is a system containing immobilized enzymes that will allow the perfect mass transfer of the substrate toward the enzymes. So this is one of the actually problems of enzymes immobilization. Then the next one, problem with cofactor and regeneration. Yes, some of the enzymes require cofactors. So it's not all of them that have the required cofactor, so there is also issue of this problem. So you see problems with multi-enzyme system. Yes, multi-enzyme system like oligomeric enzymes or multi-enzymes complex, you see that they have many subunits. So how do you now integrate the subunit on the matrix is also another a problem. Then it also leads to like loss of activity, because some of the enzymes, as a result of denaturation, degradation and instability, they may actually lose their activity. So this is the general limitation. So the next thing is the application of immobilized enzymes. So as I said, we are going to look at the application of immobilized enzymes on various aspects. One, we are going to look at it at industrial point of view. Second, therapeutic. Then third, in analytical. So like for example, in analytical chemistry, like for example, where we normally use glucometer. So what actually we do is, in trying to actually measure the blood glucose level, you know that you have to use a drop of the blood and use a strip. So actually what happened is that the enzymes like glucose oxidase is always immobilized on the strip. So these immobilized enzymes on the strip will now allow the conversion of glucose to glucuronic acids with hydrogen peroxide, and as a result of that, the hydrogen peroxide will be now monitored through the redox reactions to display the value. So it means that the higher the level of hydrogen peroxide indicates the higher level of the glucose level. So this is just in analytical technique. So you immobilize glucose oxidase, then you drop a blood and insert it into the glucometer, which will allow the measuring, or which allow to measure the level of the hydrogen peroxide and display the result on the screen that will allow you to take the reading. And so you see, this is like an aspect of biosensors, so I don't want to waste much of my time, but just the basics is to understand that you can immobilize enzymes on a script to measure it. Like for example, now currently we are under coronavirus situation, so you can decide to develop a diagnostic tools that will allow you to be monitoring the level of the viral load on a particular patient. So it's possible. because what you need to do is maybe you decide to, like for example, generally we know that the coronavirus always target the angiotensin converting enzyme, so you can decide to get angiotensin converting receptor, yes, angiotensin converting enzymes receptor 2, then you immobilize it on its particular support, which will allow you to be using the analytes, or sorry, the samples, so that you will be analyzing the level of the binding of the spike protein from the coronavirus. So that is just analytical techniques. That is one of its application. So you see, this is just the reaction. You see glucose in the presence of glucose oxide oxidase is combined into glucuronic acid and hydrogen peroxide. So this hydrogen peroxide will also be further be degraded to be releasing the oxygen level. So the level or the rate of the oxygen will be what will be monitored, so that from there you can be able to have a basic level of, sorry, the basic understanding of the level of the glucose. So in automated analysis we also use them. So this is the biosensor. So this is just like some applications of this thing. So you see, so in therapeutics we can enzymes replacement and enzymes therapies. In some conditions, like in some bone era diseases, you can decide to have like a kind of enzymes immobilized and inject it into the patients, and from there it will now restore the normal functions. So we normally use immobilized enzymes, like for example we use them like in anti-inflammatory, in anti-fibrin, that is to reduce the level of fibrin and blood cleansing, viral fighting, healthy immune system. So, and this is this. So this is the industrial application. So we can use them in pharmacy, in food industry, in productions of different types of amino acid, or in isomerization of different biomolecules into sweetening agent and many more. So that is this. So in conclusion, this is just some important of enzymes immobilization. So enzymes immobilization is the one of the most promising approach for exploiting enzyme based process in biotransformation, diagnosis, pharmaceuticals, and food industry. So several hundreds of enzymes have been immobilized in a variety of forms, including penicillin G, cyclase, lipases, proteases, invertases, etc. So research should be focused to overcome the current limitation related to the immobilization techniques, so as to expand the horizon from all round application. So ladies and gentlemen, uh, thank you for listening. So I will now just